So I'd like to, the next talk in these rapid round of talks is from Dr. Matt Bashton. Again, he's a HBBE Research Fellow, the Vice Chancellor's Fellow, and he's going to give his presentation about us dipping our toe into the world of SARS CoV-2 genomics. Over to you, Matt.
Okay, so I'm going to talk about our SARS CoV-2 genomic sequencing effort here at Northumbria University. And so early on last year, we became part of the COVID-19 Genomics Consortium, or COG UK. And this is a UK-wide consortium of various different universities, including the Sanger Institute. And our very own Darren is one of the deputy directors of this consortium. And the consortium itself was initially funded on the 1st of April with a grant of 20 million from the UK Treasury's Fighting Fund. And it reports quite frequently to the SAGE Scientific Advisory Group for Emergencies Committee. And indeed, the first report went to SAGE even slightly before it was officially funded, hence the urgency about these sort of things. And so on May the 7th, after some paperwork and formalities, we finally submitted our first batch of sequences to the project. And we've continued to, or the whole project has continued to attract funding from the government. So the Department of Health and Social Care contributed another 12.2 million in November last year. And so in total, as of today, at least internally in terms of how many sequences the project is generated across the whole UK, we've done 650,000. And in terms of the contributions that we've made in Northumbria, we've used both our GridION and NextSeq 550 Illumina and Oxford Nanopore instruments that were bought as part of the E3 funded HBBE grant. So it's enabled us to contribute to this project in a significant way. So here's the team again, and you can see Darren not against the black background, and myself next to him on the left, as well as my PhD student Matt in the white t-shirt, and other members of the lab team who worked incredibly hard to generate all of these sequences over the last 14, 15 months, I guess now. So our current statistics in terms of the number of genomes we've sequenced, we've done almost 22,000 now. And we, for most of the pandemic, were focused on what we call pillar 1 samples. So that's directly from hospital testing labs. So we're taking samples from 7 different trusts, sort of south of Teesside, North Yorkshire, all the way up to the Scottish borders and all across the west in Cumbria. And what we've done now in the last couple of months is switched over to sequencing pillar 2 samples, as we've seen the caseload switch from, I guess, from hospitalized people into sort of the community setting. And in total, we've deposited almost 15,000 sequences in the worldwide database GISAID. In actual fact, if you rank our contributions in terms of world effort, we would actually be 20th in the list of global nations. So it's not an impressive effort in terms of what we contribute as an institute. And we're now on our 630th, probably higher actually, as a result of work today in terms of our sequencing runs. So my role, what I've been doing, so I'm not lab-based, I'm a bioinformatician. And so what I've been doing is all of the bioinformatics and metadata associated with this. So been responsible for implementing all of our pipelines, getting all of our data analyzed on our HPC cluster, CRIC, and also doing a lot of quality control. We need to make sure the data we submit is up to a certain standard, as well as working with various hospital trusts and Public Health England on conducting outbreak investigations and reporting various different variants of concern. There's been an awful lot of metadata curation, which I'll come on to later. And also there's some scope for developing novel tools within this. So we have our own pipeline for processing SARS-CoV-2 genomic samples, which I've termed Arctic Slurmy, specifically because it wraps the Arctic method by sort of Nick Lohmann et al., which they originally used for the Ebola virus, which we're using as part of this project, at least for our GridION-derived samples. And it runs it on an HPC cluster that runs the Slurm job submission system. And what we've done, or what I've done, is to tack on a whole bunch of other downstream stuff onto that. So we take the variants that this method outputs and we annotate them so we can understand whether they're insertions, deletions, non-synonymous, etc. changes, and what viral orth they're in. But we also do a whole bunch of quality control, because this is amplicon sequencing. So we need to understand whether particular amplicons within the genome are dropping out. And finally, we also assign the lineage using this program Pangolin from the Rambo group in Edinburgh University, which assigns the Pango lineages, the ones that you'll be familiar with, such as B.1.1.7. We also have a whole bunch of stats that we collect for each run in real time as the analysis is underway, so we can see and feedback to the lab immediately if we're having any kind of problems. And all of this data is, of course, then uploaded centrally into the central UK sort of surveillance system, which is on the MRC Climb. So our code that we use for this is available on my GitHub page with instructions for installation. Now, of course, there's no point in genomic surveillance unless we can put a date and a place against the sample, so we can track the spread and emergence of variants and lineages across the nation in real time. And so we record quite a lot of metadata for all of these samples. In fact, there are 56 fields we routinely curate for all of these samples. So, I mean, ideally, we need the sort of location and the date, but also we need some way of linking these samples back into health records. So we have our own codes for the COG project, so-called COG codes, but also we have the original submitting lab sample ID that they used on their own tracking system. We also have information that concerns the site of where people were swabbed, the type of swab, whether we got an extract or lysate, etc. Other stuff such as out of postcode, what hospital they were admitted to, or the sample was taken at, whether they've been admitted to ICU, whether they're a healthcare worker, whether they're a care home worker or a care home resident, things like that. And we also collect a lot of details about the PCR test. So throughout the pandemic, a number of the commercially available PCR tests have failed on certain probes because of extra insertions and deletions that are present within the viral genome. And we're managing to keep track of all of that, as well as the performance of these different platforms, by curating quite detailed and rich metadata. And we also log all of the metadata connecting to our sequencing run and bioinformatics pipeline. And so all of this metadata is ingressed centrally within MRC Climb by a system called Majora. And it's essentially a continuous integration system for dealing with I guess, what they term sequencing artifacts. And here you can see the quality control that one of our samples has been subjected to centrally. And it's also then further been submitted automatically into GISAID and in European Nucleotide Archive as well. So that's sort of the crux of what we're doing on our end. But some of the more interesting things that we've done is to run outbreak investigations. And there's an excellent tool which was being developed by the Ramo group at Edinburgh University that over the course of pandemic we've worked with and fed back on in terms of its design and how it operates, called Civic, Cluster Investigation and Virus Epidemiology Tool. And this takes advantage of something that we have centrally within COG, which is a UK-wide, if not global, phylogenetic tree, which is updated on a daily basis. And what this tool does, which is essentially an absolute game changer, is if I have, say, 10 patients who I think are part of an outbreak within a hospital, I can submit those 10 patients with some associated metadata of my choosing into this tool, and it will then align those sequences into the global phylogenetic tree that's generated within COG. And the real power here is that if those 10 patients are part of the same outbreak, then they will map into that part of the tree. But if there are other patients in the hospital that have perhaps been moved or are in different wards, purely by virtue of sequence homology, it will also route those, even though I didn't know about them, into the tree. And in fact, this technique is so powerful where we see it pulling in, or we've also included within our query sequences, hospital workers. What we find is that where the spouse of the hospital worker's sample was collected by Pillar 2, because they also were concerned about being positive, and that was sequenced by a different laboratory using different sequencing hardware, we find that it will routinely resolve people's co-residents into sort of unique little cliques within the phylogenetic tree. So you certainly can't track people by vaccinating them, but actually it turns out you can track people once they acquire COVID infection, at least by their viral sequences anyway. So we've done an awful lot of outbreak investigations using this tool, mostly in the hospital setting. And what I've got here is an output of one of these phylogenetic trees. I've had to redact an awful lot of this plot because it's somewhat sensitive. And what you can see here are the individual sequences represented by the sort of red dots on the left within the phylogenetic tree. And then we've got other information encoded in colors. And what this is actually a representation of is an outbreak of coronavirus within a prison. And what's really neat about this is in the final column to the right, what we have is a colorization, which is actually the wing of the prison. If you map that across the left, what you can see is that the sort of phylogenetic branching of this tree precisely correlates with the wing of the prison. That means that in each wing of the prison, one initial incidence of COVID was sort of acquired, and then it spread around that wing. And we can also see where we've got a red dot in the middle column rather than a blue dot, a prison warden. And what's interesting is it has, via Pillar 2 in homology, pulled in another sample, which we're following up to see whether there's someone who cohabits with this warden. So this technique is immensely powerful. And I think it's a real game changer in terms of investigating outbreaks. And we've routinely fed back into hospital infection control teams on these natures. So we've also been involved in this trial, Hospital Onset COVID-19 Infection Clinical Trial, or HOKI for short, which is a similar kind of thing to Civic, but it used different tools. This was led by UCL. And here what we did was to collect the same additional metadata that concerned where patients had been around in various wards. And we had sort of two phases of this project: a slow phase where we had to acquire the samples, sequence them, and then produce this infection control report within 5 days, and then a rapid phase where we had to turn all of that around within 48 hours. So the treatment in this clinical trial was genomics. And we certainly did make a difference in terms of feeding back to infection control teams and then be able to implement recommendations and changes based upon our findings. And so come more currently to the problems that are facing the UK. And currently we have a really high incidence of cases due to what has become a particular descendant of a particular Indian lineage, otherwise known as Delta. And so the earliest sequences of this really emerged in September last year, of this particular lineage, at least in terms of sort of hereditary origin, within India last year. And in the beginning of February, India had a relatively low case rate of just 9,000 cases a day, but that rose to 68,000 by the end of March, which admittedly was quite a shocking rise at the time. And the first few cases we saw, at least within the northeast of England, that we sequenced within a rapid 24-hour turnaround with travel history from that continent, began to start arriving in the beginning of April. And so these were people who had either traveled or one of their family members had. And really at the time, there were very few sequences that existed with, from this lineage. There wasn't a lot of sequencing underway in India. And it was interesting because at the time, the defining stem mutations of this lineage were really L52R, which had been actually observed independently within a Californian lineage, P618R, and sometimes E484K, which is also sometimes present, which is also present in the sort of South African lineages. Worryingly, what we'd sequenced had a whole bunch of different spike protein mutations. It had this deletion, 156, 157. It didn't have E484K, but it also had this additional T478K and a D614G, which had previously been observed in a Spanish lineage that spread around Northern Europe as a consequence of mostly holidaymakers last summer, and as well as another spike mutation, D950N. And so we actually sequenced both of these samples twice because we were so concerned that this result wasn't correct. And at the time, there was a big discussion thread going on within COG over Slack about where these mutations had come from and what was going on, because really the sort of sequence space had been somewhat underrepresented from these lineages. It then turned out a couple of days later, after I was pained to sort of point out that ours were different, that what we'd sequenced was probably about the second case ever of Delta within the UK that had hit pillar 1. And so that was really interesting. And so the Indian lineage itself is forked into 3 different sublineages: 617.1, 617.2, which is later been termed Delta by WHO, and 617.3 as well. And so here you can see a graph of how the various different lineages have sort of spread around within the UK. And what you have in this sort of greeny color in the middle is the Kent lineage that was popular over Christmas, starting out in sort of September last year. And you can see that as of April, the gradual introduction of, by virtue of travel from the Indian continent of the Delta lineage. And what's interesting is that came back and was introduced at the same time as Kappa, the 617.1 and the 617.3, which doesn't have a designation from WHO. But there was then a subsequent meteoric rise, really, of the Delta lineage. And you can see that it actively displaces the sort of Kent or Alpha lineage really quite rapidly to the situation we're in now, where it's accounting for well over 90% of all the cases in the UK. And this is, of course, data from the whole of the COG project, not just our sequencing. So worryingly, the concerns obviously are about transmission. And so we know that the Kent or the Alpha variant or lineage, shall I say, was 60% more transmissible than the original strain of the virus. It now transpires that Delta is about 70% more transmissible than the Kent one. And so this is really quite a very transmissible virus. And in fact, in Sydney, where they have excellent contact tracing, where they follow up cases with CCTV as well, what they've been able to see is that fleeting interactions of individuals within built environments, where they simply pass each other for 5 or 10 seconds, is enough to confer transmission. But the other concerns we have with regards to Delta are really immunogenic escape, and work by Ravi Gupta, has showed that and this is a preprint which is under consideration within Nature. This is not our work, this is his, but it's shown that basically it manages to escape both sera from individuals who've been exposed to the first wave within the UK, and also to some degree there's a 9 to 8-fold reduction in the antibody and its sensitivity to antibodies from both of the two main vaccines. And so this brings us on to what's happening now, and what we can expect to potentially happen and what we need to worry about. So one of these things is Delta Plus, a lineage. It's a descendant from the 617.2 termed AY.1. Delta Plus is kind of its name in the media. And this has the same spike protein mutation as one of those that exists within the South African lineage. So this appears to be some kind of convergent evolution in terms of the fitness landscape of the human beings that it's trying to infect. Thankfully, at the moment, there are only 52 cases of this that have been reported within the UK, but this is precisely why we need genomic surveillance. And so, sort of getting to the end now, but we've recently received, as part of a package that I put together of proposals, 40k to fund a postdoc from COG in collaboration with a former colleague of mine, Rafael Nabshmanovich, at the University of Montreal. And there's two main prongs to this. So one, we're concerned about the ability to still accurately genotype Fox and Thuis within samples that have low CT values. It's perhaps less of a concern right now that we have a sort of high incidence of Delta. But the second is what I've termed a kind of spike protein anti-malware scanner, which is an ability to annotate and also prioritize variants, particularly when the sequences turn up on our from travel on our shores, etc., so to speak, without much of a background knowledge of what's going on. So the idea is that there are a rather large amount of studies now of various interactions of the spike protein with monoclonal antibodies and sera from patients and things like this, as well as computational simulations about the conformational stability of the virus. The idea is to take that wealth of knowledge and use it in a tool which can actively map these cases as they're filtering through genomic surveillance networks like COG, for instance. And it's of particular importance now that these things are being done nationally on a large scale by centralized things, rather than there being a community of people looking over the results by hand, so to speak. So just to acknowledge the wider team within COG at Northumbria University, I've highlighted everyone there who's an HBBE member, also the funding of COG and the funding of HBBE, which has allowed us to contribute to this project.
Thanks, Matt.